Our next speaker and the last speaker before the intermission is Dr. Ole Andreasen, and he is being feted this year after having won the Colvin Prize. You can come on up here, Ole. Ole has a position, I think that you've been the farthest coming to New York, but I didn't even add up the miles. He is a professor of psychiatry at the University of Oslo in Norway, and he's the director of the Center for Precision Psychiatry in Norway. He has many roles and much input into our field through the PGC and through the Enigma Imaging Group. Ole might be best known for translational research in bipolar disorder, and that's, of course, what he won his prize for from this year from BBRF. He relies on molecular genetics, brain imaging, and cognitive performance to identify pathophysiology in bipolar disease. He chairs the PGC section on bipolar disorder and the Enigma imaging section on bipolar disorder. The contributions to neuroscience that Dr. Andreasen has contributed are so, I don't have enough time to really say them all. I'll just say that he's identified distinguishing neural signals in bipolar disorder between bipolar disorder 1 and 2, related to specific cell types and molecular mechanisms contributing to personalized medicine. And I hope we hear more about that today. I have to say that Dr. Andreasen publishes prolifically in the scientific literature. It's not all about bipolar disorder. It's about, in fact, if you look at his most prominent publications, a minority are from bipolar disorder. But there are also many in Alzheimer's disease and some in just regular brain mechanisms. He's published it, I'm just going to say, in the CV that I read, he had 1,232 publications that gave him an H index of 162, something I've never seen before. It would be easy to say that Dr. Andreasen does not confine himself to bipolar disorder, even though he has a long-standing reputation in this area that we'll hear something about today. Thank you.
Thank you. Thanks for a very nice introduction, and also I'm very thankful for the Colvin Prize. That's really a big honor. And I will today start with talking a bit about the genetics. That is a tool I've used a lot to study bipolar disorder mechanisms and also how we can translate it to the clinic. But heritability, that is the concept that you inherit genes from your parents that play a role in traits and diseases. And this is a picture to illustrate this concept. They are identical twins, then they were born on the same day, but they died on the same day, and they had the same disease. And they also shared many other aspects of life, as you can see here. And the question is, even though then they have absolutely 100% the same genetic variation, they also shared the environment. You know, grew up in the same family, they had the same neighborhood, the school. So to figure out what is genetic factors and what are environment, you can do twin studies. And then you study identical twins with fraternal. Then they just have 50% of similar genes. And then you can see what is the rate like bipolar disorder in fraternal versus identical. And here you can see that bipolar disorder has really a high heritability, one of the highest among the common diseases of humans. And this then we can use to study, the mechanisms of the disease, but also see if we could translate it to the clinic. But it's just to show that the heritability here is over 75 out of 100. That is not the same as individual risk. That is, for bipolar 1 disorder, is like in the population is 1%, and if you have a first-degree relative, it's like 10 times higher. So it's not like 75%. So that is very different concepts. Anyway, so what we then can do with these studies is that we can figure out what is the heritability made out of. And then the theory was that there are many, many genes that work together to increase risk of bipolar disorder. And then to find them, you need really, really big samples. And that's why you need this international consortium, and that we are working in this psychiatric genomics consortium, where we can have a global collaboration to identify or analyze the genetics of psychiatric diseases. And now, due to technology, it's so cheap. You know, it's cheaper to genotype your DNA than park at the hospital. At least, I think that's the same in Norway and at Manhattan. And that can enable, then you can have really big samples. And we can then study which of these variants are associated with bipolar disorder. And what we're then doing, you know, this is a DNA, the different codes here, like adenine, thymine, cytosine, guanine, and you see which of these variants are more associated with this disease. Like here we have a big group of patients and then healthy controls, and then you do this genotyping of all the different samples, millions of variants. And then you compare them in a statistical analysis. And this is called a Manhattan plot. So each variant here, you know, we are here on Manhattan. Now it looks like the skyline. So each little variant here is an association of this gene variant with bipolar disorder. And you can see now they're like, if you count them across all the different chromosomes, all the black dots here, that's the variants. It's really highly associated with bipolar disorder. And there are like 300s now, and more are coming. But why do you need the skyline? You know, that's not helping you. So what we can then take these variants and look into mechanisms. Can we then use all of these new variants to see, are there any new mechanisms of bipolar disorder? And one thing, I'm just highlighting a couple of examples here, is many of these variants were associated with these calcium channels, you know, that are involved in transmission. You know, we have, 100 billion neurons, each are associated or talking to 1,000 others. And these genes of bipolar disorder seems to be associated with some of these like channels that are helping transmission between neurons. And these are the voltage-gated. And that could be a little or new opportunity for developing new drugs to sort of stabilize the transmission. And then you can do all of these genetic variants from the Manhattan plots and link them to large repositories where they have genotyped neurons in the brain. Then we can see, well, it's not every part of the brain that's associated or enriched by this signal here. It is amygdala. And I think we'll hear a really great talk later today about how amygdala is related to emotions. And the same with the cognition. You can see here there are enrichment of these genes in hippocampus. So this can really sort of form the foundation for more in-depth studies of these regions. But then, can we then translate this? So I see patients 1 day a week, and I'm really thinking, we don't need just genes, and you don't need papers in Nature or whatever you need to translate these findings to help patients. And how can we do that? Well, that's called precision medicine, targeting the right treatment to the individual patient. Then we need information from big data, from genomics here, combine it with electronic health records and big, large cohorts. And this is what we are trying to do with then the translation of these findings. So there's so many variants now, so you can combine them and see if, it's called the polygenic score, just sum up all these variants and see how are they related to the disease. And maybe there could be some of the, like a distribution. If you're over a certain threshold, you're more likely to have susceptibility, and then environmental factors can sort of lead to disease onset. And and this is what we then can do in something called polygenic risk score. And that explains for bipolar 1 now, 18%, and for bipolar 2, not that much. And why this is promising, it looks like it's possible then to predict disease, but I just want to warn, this is not yet clinically relevant. It's too much variation here and also too little, but the concept that you can use, maybe identify subgroups, or maybe people who are more likely to respond to a certain treatment by using these tools. And then you can, since we now have millions of participants in these studies, we have so much data. There were like 2 million cases and 150,000 cases and over 2 million controls. So we have so much data here. And these are all these millions of variants associated with bipolar. And here we compare them with depression and can find then with some machine learning tools which are overlapping. And then you can estimate the number of variants. Here we can compare with depression. That is more like lots of variants, over 10,000. In bipolar, they're like 8,000. And then you can see there's a lot of overlap because they share many disease characteristics. But these that are specific for bipolar disorder, that's like over 1,000, they're probably related to the manic phases of bipolar disorder. So this is something we are working on also to link it to the brain. And you can see here there is also overlap between bipolar disorder, all the genes here, with cortex, how the cortex is developed. And this is why brain imaging is also now something you can put into a machine and calculate automatically. This is something called FreeSurfer, and you can identify the thickness of the cortex, the different structure, and also the cortex. And there's now thousands of these scans out there we can use in research. And that's what we're doing in the Enigma consortium, where you have brain scans around the world. And then here is a picture of the cortex of bipolar disorder, and you see it's very, very small effects. These are the yellow, is tiny, tiny, tiny effects, or the difference between bipolar and controls are very, very small. And this is not seen if you are treated with like lithium. So this is, again, here, it's many, many small variants that plays a role. But then we can also see genetic overlap between not only diseases, but also human traits that are highly beneficial, and that's creativity. And here you see an example of this polygenic score for bipolar that's associated with creativity, creative professions. And we also showed an overlap between bipolar disorder and cognitive performance. In some parts it was improved, the genetics of these factors. And I think this really talks to the well-known increase in authors and artists and creative professions in bipolar disorder. That seems to be, in fact, the genetic underlying mechanisms here. So where are we going now? It's a lot of opportunities in this field, I think, especially we're using these new methods where we do large-scale analysis, because I don't think genes explain, we are focusing on genes, but they cannot be alone. You need to have genetic variants, you need to have brain imaging, you need to have environmental factors. Everybody knows that stressors in the environment can trigger a new episode of bipolar disorder, and we need to understand the hospital records, how that can help in understanding the disease, and also omics, like we can measure now all kind of things in the blood, and putting them together in like a multimodal, meaning that you use data from all kind of different aspects, not only focusing on one measure. That can we have so much data now, as millions of samples, that could help with better stratification or prediction models. When will you develop disease? What will the outcome be? Or also to improve the diagnosis and also find better treatment for individuals. And this is what we are really working on now. And then I can just highlight this is a picture of the Scandinavian country Norway, and I live here. And we have a lot of data there from the healthcare and from big biobanks that we can follow people over time. And we're now working on developing tools that can sort of put all of these together, all the biobanks and population surveys into some kind of a training opportunity to develop tools that can make us understand better the causes of these diseases and also the outcome. And especially the problem with doing a lot of research in hospitals is that you don't understand what happened before people became sick. So in Norway, we have also something called the MOBA. It's a birth cohort of 100,000 kids followed over time. They have the genetic risk factors, the genotype, and then when you grow up, there could be all kind of environmental stressors. And then you can study who have genetic susceptibility, who had the different environmental factors, and who developed mental illness, and who were resilient, that did not develop disease. I think this is then opens up for more opportunities for prevention, but also to understand why people develop disease, because there are many out there that have the susceptibility, but they manage not to be sick. So I think there are big opportunities in this area of using these kind of longitudinal data from large cohorts. And I think that's enough for the future. I also want to thank my team and everything, but what we have shown here is that the genomics can really give new insight into mechanisms, but also these calcium channels I want to highlight because that's a new way of looking at new drug treatment. We saw that the hippocampus and amygdala are specifically involved in bipolar disorder mechanisms, and also if you do brain imaging like in the ENIGMA study, it's not like a big change. There is small subtle changes that are more dynamic. And then the clinical transition to how can you bring this into the clinic. I think it can be used to differentiate bipolar disorder from depression. It can also form the basis of this: who will benefit from a treatment and who will not benefit and only have the side effects. And then finally, there are some potential of this polygenic risk score, but it's still very, very preliminary. That's more like a potential. It's not clinically useful. So here I just want to thank all the members who have been working on this. This is really team science. A lot of these international samples are supported by different European and American funders. And also, I want to thank all the study participants. Like 150,000 contributed to this study. Thank you.
Thank you, Dr. Andreasen. It was a wonderful talk. Thank you. And under time. Questions? Yes, please. One question, one person.
I work with borderline personality disorder, and I work with borderline personality disorder, and almost every person with borderline that calls our helpline is diagnosed first with bipolar because of the differences. Seeing a mood switch, they assume it's bipolar. It results in tremendous damage in the long run. Are you doing anything that can help the field to distinguish between diagnosing bipolar and borderline personality disorder for the sake of the people who suffer the consequences?
Yes. So that's a great question. So what we are working on in this big international study, they are trying to find genetic factors for personality disorders, and then we can use them to compare with bipolar and to see it's not happening tomorrow, but this is really, if they have different genetic signal, we will manage to discover that, and that can be used in the clinic. But you need to tweak it a bit, and you need to do this machine learning kind of novel tools. But that is really, really what's ongoing. So very good suggestion.
Can I follow that up with a question, Ole? That we started out a century, 2 centuries ago with psychiatric diagnoses that were diagnoses based on behaviors mostly. Now we have all of the elements that go into precision psychiatry. Are we going to have different diseases in a couple of decades?
It depends on what we want. I think today I'm also working in the clinic, and I find our differential or the different categories sort of useful. But I don't think they will maintain, because if we find subgroups, there could be a cross-diagnosis, it could be a subgroup with bipolar and some personality traits that need a specific treatment. And if we learn about that, I think that then you can really categorize that in a different way. Or there's a lot of overlap between like severe bipolar disorder, they have psychotic episodes, and maybe that is more like psychosis spectrum that needs a different category than bipolar or schizophrenia. So definitely, and I think that I was at a meeting here now talking with the new APA, or we call it DSM working group, and I think they would like to add some of these factors now in the new categories. But what I'm saying is it should work. If it doesn't help us, why should we change? Why, I like these or I like that. If it doesn't help us in improving the treatment or prevent, then a couple of genes won't help us. You know, it has to be clinically relevant.
Any final questions? Yes. It's coming.
How have you seen AI advance the science, especially with all of the subjects you're able to get information from?
So it's a wonderful opportunity now, because we have been working in this big data field for a long time, and now we can have these new models that really have so much compute power that you can find new non-linear effects. So all of this is based on case control, one at a time. But if you have multiple factors, it could be that this gene is associated with this gene, and if you then have this specific environmental factor, we're not able to find those special cases now, but AI will do it. But then you need to put it in a safe, you can't give it to, well, you need to be secure and not give it away to people or companies or whatever who don't take care of patient integrity and the data ownership. And this is what we are trying to set up. You need it on Norwegian soil and put the data together.
I'll ask again for a last question from anybody. If not, we'll contribute these 3 minutes. Oh, well, we have one more question. I was just going to say you could have a longer coffee break.
Oh, thank you. I'll make it quick. I apologize. I work with bipolar patients, and a lot of them come to me misdiagnosed and mistreated as depression. From clinical perspective, some of your thoughts, what can we do to avoid that? Because a lot of times they come with already experiencing manic episode due to antidepressants.
So,
thank you.
Yeah, the question is about how to differentiate unipolar from bipolar depression. So I think you need other type of treatment for bipolar depression than unipolar in general. But if you only have depression episode as your first sign, it's impossible to know if that is part of a bipolar than if it's not part of unipolar disease we are seeing, because we haven't seen any, we have not had time to develop manic episode. And that is exactly, I tried to show with my little comparison between depression and bipolar, that there could be distinct factors that are overlapping and that are different. We're now working a lot on trying to differentiate this and find those specific markers that could differentiate, because then you don't need to wait until you get the symptoms. That is the point. You can do these tests early on and then treat as if you already know that they will have manic episodes.
Thank you very much, Dr. Olaisen. We will start the coffee break.